All right, so welcome to Unit 2. In Unit 1, we talked about the basic characteristics of life, some of the chemistry, and then some of the cellular things. So now we're going to go actually into the cells specifically, and we're going to be looking at what's going on in the cells, how they're getting energy, how they're doing homeostasis, and some of the details that allow us to continue to better understand organisms as we move through this semester. So today, Chapter 5 is a little bit of an introductory unit to several different topics that will continue to be important. And so each of these is going to be broken down by those check your understandings. I do strongly recommend making sure that you understand everything before you continue on into chapters 6 and 7, which are photosynthesis and cellular respiration. So today, and you'll see that there again is a lot of chapter objectives for this, but these are definitely going to be talking about energy as well as enzymes and then how cells are maintaining homeostasis. And so while these seem like a lot, I promise they're not that much, and we're going to go through each section. And as always, if you need more help with any of these particular objectives, you should go and read the textbook corresponding sections to these. These are from the textbook directly. So let's start off by talking about energy. When we're talking about energy in a biological system, what we're really talking about is ATP. We're talking about cellular energy. But in order to fully understand cellular energy, we need to talk about energy processing from this idea that solar energy comes into our ecosystem, is captured by our producers that do photosynthesis, our plants and algae, and then is utilized by any sort of consumers. At each step of this conversion process, energy is eventually lost from the ecosystem as heat. Now, it's not lost in that it's just disappeared or destroyed. Instead, it's just a byproduct. Like when you run your car and your hood of your car gets really hot, or when you do a workout and your body starts heating up, you are converting that energy from potential energy, that stored energy in your body as that glycogen or that fat, into kinetic energy. And during those conversions, you do lose some energy as heat. In this case, it explains this idea of energy processing, that energy is continually needing to be added into our ecosystem. It is not going to be cyclical. It is one way. It is a flow. There is a flow of energy. Once it is lost as heat, it is back into the atmosphere and it is going to be released, okay? However, the nutrients that we get, those carbons and hydrogens and oxygens, are always cycled through these processes. So in this way, we are learning again that energy flows and nutrients are cycled. And I had mentioned how your cells are the best recyclers out there. Well, the energy that your cells use is called ATP, adenosine triphosphate. ATP is a type of nucleic acid, adenosine being that adenine. It's kind of a base off of there. Now, ATP is the energy currency for cells, and it's really pretty simple. It is consistently converting from ATP to ADP plus P, because what happens here is we have three phosphates jammed together off of this ribose with an adenine. These three phosphates are not happy. I like to think about it as if you've ever been on a school bus with like middle school aged boys who are probably too big to sit three to a bus seat, but they cram on there anyway. We know that is an untenable situation. Eventually, the person who is at the end near the aisle is going to get bumped off or pushed out, and those boys are jostling and they're kind of trying to squeeze in. That's what's happening with these phosphates. So ATP is not going to be a very stable long-term energy. It is something that is used to carry out those activities, and it is utilized basically as quickly as it is made. However, once it's used, it's not wasted because, again, nutrients and things in your cells are cycled. And so that adenine and ribose that has two phosphates, or ADP ADP, that's right, instead of ATP, adenosine diphosphate, rather than adenosine triphosphate, is reused. It's a continual breakdown and regeneration process. And this is going to be a theme in cells when we're talking about nutrients or things that are built off of like nucleic acids or carbohydrates, okay? What's really cool is your cells use so much ATP just sitting. So when you hear about needing a certain amount of calories just to maintain normal body function, that's what they're talking about. Your cells are using this ATP so quickly, even if you're just sitting, even when you're sleeping, just to keep your heart beating, your lungs moving, your brain functioning. And what else is really cool is ATP is only used by the cell that makes it. So ATP can't be made and then pushed to another cell. It is just continually used within that cell itself. So it's going to be really useful for biological energy. And ATP is another one of those molecules that is conserved across living things. Bacteria use ATP, you use ATP. This is going to be one of those things that is just really key to life on this planet existing. Okay? I've mentioned many times in just the last, 5 minutes here, that ATP is going to be part of that energy flow. So remember, energy is going to flow and it is either used or lost as heat. Okay? Sunlight comes into our ecosystem, into the chloroplast of our plants and algae, and that chloroplast can convert that solar energy into glucose. So it is taking solar energy and converting it into chemical energy. The chemical energy in glucose can then be used by the mitochondria in eukaryotic cells to be generating cellular energy in the form of ATP. Okay, so cellular respiration and photosynthesis are opposites of each other. They are continually cycling the nutrients between the two, between chloroplasts and mitochondria. And that's how when you eat food, you get the energy from that food. That food all begins with plants. Even if you eat a steak, the steak is from a cow which ate plants. And so we are going to be seeing that these nutrients that are cycling are always there, but energy is lost as heat. Okay? So that is our first part on cellular energy. Make sure that you can check your understanding. Make sure you can answer all of these and try to draw yourself a diagram illustrating the pathway of energy flow. Be sure to label those appropriate types of energies and the organelles where appropriate. Okay? Once you've completed your check your understanding, let's move on to enzymes. So enzymes are a protein. Enzymes are a protein that facilitate reactions that are happening in the cells, such as making glucose into ATP and taking that glucose and converting it into ATP. Okay? Enzymes are responsible for your metabolism. Metabolism is the sum of the controlled reactions that happen in cells, and these enzymes speed up these chemical reactions, okay? What's really cool about enzymes is they're not used and wasted. Again, your cells are the best recyclers out there. Instead, each enzyme is going to do its job and then be cycled again to do its job the next time it encounters that particular substrate. Enzymes are specific to their substrates. So when I talked about if you put starch like a saltine cracker in your mouth and you hold it in your mouth, you start that reaction of digestion in your cells of your mouth. And what's happening there is your cells, you have glands in your mouth, your salivary glands, that produce an enzyme called amylase. Amylase, okay, is going to be the enzyme that starts to break down that starch into its sugar component parts. So if you hold a saltine in your mouth for long enough, it will start to taste a little sweet. Now, I'm a little grossed out by the texture. I'm not going to hold a saltine in my mouth that long. But if you do, you are breaking that large polymer of starch into its component parts of glucose with the help of an enzyme called amylase. What I want you to notice is it doesn't matter if we're doing carbohydrates or fats or proteins. The majority of our enzymes end in ASE. So it's like our sugars ending in OSE. So we have things like lactose. Lactose is broken down by the enzyme lactase, right? So what we're seeing here is that there is an enzyme specific to lactose. And I like to bring this one up because people are always curious about how they become lactose intolerant. Well, lactose intolerance is actually the normal state, and lactose tolerance is a mutation. And what happens is, because we are mammals, we do rely on milk that has lactose in it when we are younger, when we're babies. And as we get older, we don't need that milk product anymore, but humans like to drink milk continually through our lives. So if you are able to handle lactose as an adult, it means you have a mutation that continues to produce lactase. So your body continues to produce that lactase. But most people actually are lactose intolerant because they don't continue to produce that enzyme lactase into adulthood. That's why if you drink like the Lactaid milk, it's actually going to taste maybe even a little sweeter because they've already added in the enzyme lactase to start breaking down that lactose. Isn't that just cool? And if you want to get oils and fats out of clothing, you have to add a lipase in order to break them down. So because oils and fats are nonpolar and hydrophobic, and they just avoid that water. Okay? Enzymes are fascinating. They're really great cleaners if you have an enzyme specific to that substrate. And let's talk just a little bit more about how enzymes lower activation energy, okay? What ends up happening here is that the products that we will see from a reaction will occur with or without an enzyme. However, with the enzyme, those reactions are more effective and happen to occur with less energy, which also means that they tend to go faster or be easier to accomplish. So we have an image here where we have a figure of a reactant, and we see potential energy is really high without the enzyme. This would be if you were drinking lactose without having lactase, you'll break some down, but it's not going to be effective, and that's why you end up having that gastrointestinal distress that we tend to see with people who are lactose intolerant. With an enzyme, you do break that reactant down into its product, and you do so more effectively. Another way to look at this, and my favorite way to look at this, is if I were to build a birdhouse, okay? I can build a birdhouse using a hammer and nails. I can do it. It might take me a while, and it takes a lot more energy to hammer a nail than it would if I used a nail gun. That nail gun is making the same product. I'm making a birdhouse whether I use a hammer and nails or whether I use a nail gun. But with the hammer and nails, it takes more energy. And so that's similar to a reaction happening without an enzyme. I'm still going to get the same product, but it's going to take me longer to get there. At the same time, if I use the nail gun, I might be able to make 3 birdhouses in the time it takes me to make 1, assuming I have all of the wood already cut out and prepared. And so with that enzyme, with that nail gun, I am able to actually speed up my production of that process. And in doing that, I am seeing more effective use of the energy that I actually am using. So enzymes are going to work in that way. They are lowering activation energy. Now, I've mentioned already that enzymes are very specific to their substrate. What that means is enzymes have something called an active site. That active site is only going to interact with a substrate that matches. We call this a lock and key. So if you put the key and you try to put a key in a lock that doesn't fit, it will just not go in. That's going to be the same sort of thing with our enzymes. Now, there might be substrates that are similar that can fit on the enzyme, but if you put a key in and it doesn't turn, it might fit, but it doesn't unlock the lock. There's no reaction that's going to occur. The door won't unlock. And the enzymes, same sort of thing. If there's a substrate that fits the active site, but not perfectly, we will not see a reaction occur. As such, this enzyme is going to take in a particular substrate, and then we see the reaction occur that lowers that activation energy, and products are made, and the enzyme can be reused. Okay, this is fantastic because we don't want enzymes just running around willy-nilly, breaking down whatever they come in contact with. If I have the enzyme lactase, I don't want that enzyme to start breaking down lipids. That's not effective. It's not going to be the best use of that enzyme, and I don't know what it's breaking down, right? It could start breaking down something that I don't want it to. So instead, we see this induced fit model. What's really cool about this induced fit model is this has been used to identify the steps of metabolism, like what we see in photosynthesis and cellular respiration. We know that if we take out a certain enzyme, if as a scientist I go in and I block that enzyme, the rest of the reaction will not occur because that enzyme in that step is making the next step capable of occurring. And so I'll see the speed of reaction downstream from that enzyme stopping or slowing down. And so that's when we can see like, oh, lactase is the one that's helping you digest lactose. We can then make food products that have lactase already in them to aid the digestion of those dairy products. Okay, so enzymes are specific, but when an enzyme is in a cell doing its job, it still needs to be controlled more than just being specific to a specific substrate. And as such, we do have a lot of different kinds of enzyme inhibition. Again, enzymes are generally breaking or making things. And if you've ever tried to build, especially if you had younger siblings, if you ever tried to build a card house or blocks, and you had a younger sibling who came through and knocked it down every time you got close, you get really annoyed. Yeah, same sort of thing with enzymes. So there's a lot of different ways that enzymes can be controlled in a cell. We can have different feedback inhibitions where when there's enough of a product that's being made, that product then blocks the enzyme from functioning. There's inactive pathways where your genes will actually be turned off if you're no longer needing that enzyme, such as what happens with lactose intolerance. Or we can have inhibitor molecules. I would like you to know about the two inhibitor molecules and understand that this is not the only way that enzymes are controlled, okay? The two kinds of enzyme inhibition that I want you to know about are competitive and non-competitive inhibition. So inhibition is a stopping or a pause of a process. This doesn't destroy the enzymes, and those enzymes can then be reused, which is great. We want that to happen. In competitive inhibition, the substrate is blocked from binding to the active site by another molecule that's similar, but that does not activate that enzyme's process of breaking down that reactant into a product. Remember I said that some things can fit in the active site, but it doesn't turn the key? Competitive inhibitors do that. I like to think about this as if you were to go and park at the mall. Okay, you've ever been to the mall in Jacksonville when the Marines have a day off? Most of us who live here for any period of time determined not to do that. But if you were to go and try and park at any sort of parking lot and you see a space that seems open and you're like, cool, and you go to swing in and someone has a motorcycle parked at the end of that parking space or a small Fiat that you couldn't see because of the large trucks that were blocking it on either side from your view. That is competitive inhibition. You cannot park in that parking spot. Somebody else is already there. That's what we see with enzyme inhibitions. Okay, so competitive inhibition is a direct blocking of that enzyme's active site. Non-competitive inhibition is a little bit different. What that is actually doing is there is a secondary site on the enzyme called an inhibition site, and there can be an inhibitor, a non-competitive inhibitor, that binds to that inhibition site. In doing it, it basically changes the shape of the active site to such a place that the substrate might be able to bind, but the reaction is blocked. Same thing with the parking lot. You go to park and you can squeeze into a space, but if you can't open your doors, it's not helpful to you, right? You can park in that space. Someone's parked over the line a little bit, or they have those huge trucks parked in the compact parking space. You might be able to fit in there, but if you can't get out of your car, your reaction is blocked. So we're seeing the same sort of thing in non-competitive inhibition. All right, keep in mind enzymes are really important. They are proteins, and make sure that you can check your understanding on these 5 questions here. Once you've done that, go ahead and continue the lecture into cell transport. Okay, let's talk about cell transport. So cell transport is going to be really kind of cool. So this is a part of homeostasis. And homeostasis is going to be allowing cells to maintain that internal and external environment. If you remember, all cells have a plasma membrane, and that plasma membrane is controlling what can move into and out of cells. And I don't know why my little GIF, oh, there it goes. Okay, I had to click it. Why my little GIF wasn't working. So, the Amoeba Sisters have this fantastic illustration of this cell transport. So there are a couple of ways that cells can move substances across the membrane. Passive transport does not require energy, and generally speaking, substances like water or salts will move from higher to lower concentrations. It's going to be a very simple process. It's going to be something that we call diffusion or osmosis, or even facilitated diffusion, and it doesn't require any energy. Okay, but if I want to move something from a lower to a higher concentration, I'm going to have to add energy into that. Okay, or I might even have to do something called bulk transport, where I am actually physically pulling it into the cell. And I mentioned how we can build those little vesicles in cells from the cell membrane, and then maybe that bonds with the lysosome to break down, or it might then be utilized. For instance, your hormones are often too large to just move across the cell membrane on their own, and do rely on that bulk transport to move them into the cell that needs that hormone to start whatever the next process is in that hormone pathway. Okay, so these different types of cell transport, we have to keep in mind that cells, again, are like a fence. Your cell membrane is like a fence. Your fence is there to keep some things in and to keep other things out. Okay, we do not want just anything moving in and out of our cells without any sort of control. We want some control in order for that to happen effectively and to maintain that homeostasis, that internal regulation. So let's look at the plasma membrane and talk about selective permeability. Okay, so again, your plasma membrane is made of a lipid bilayer, a phospholipid bilayer, which means that we have polar heads on either side, inside and outside of the cell, with a layer of non-polar tails that are going to potentially repel some things such as charged molecules. In addition, these are not going to let everything just move across easily. Selective permeability allows for some substances to pass freely and others are not. I really do like the idea of a chain link fence when I'm trying to think about selective permeability in my plasma membrane. Now, think about a fence that's going to be around a chicken coop, okay? I want small little circles. I want a tight link in my chain link, that's going to be my chicken wire, around my chicken coop. I don't want my chickens getting out, right? I don't want my chickens getting out, especially when I live near the woods and there are things like raccoons and foxes. And I don't want those raccoons and foxes to easily get in. But I need air to pass through, right? If I don't have air passing through to my chickens, they're going to die because we need air. I also don't mind if maybe some bugs and things pass through, because those insects are going to be eaten by the chickens, which is good for them. And so I want to have the right size fence to allow for certain things to pass through unhindered, but to stop other things. Now, maybe I let my chickens free range during the day. Okay, a lot of people do that. That's cool. I'm going to have a gate that I can open that is going to act like our active transport, or even our facilitated diffusion, where I have a molecule, in this case a protein, that is letting them move across that membrane. So if I look at my selective permeable lipid bilayer here, this image, small non-polar molecules, meaning that they just overall don't have enough of a charge to be repelled, are going to move easily across the membrane. And that's good, because I want things like oxygen and carbon dioxide and nitrogen to easily move across that membrane. I need oxygen to do cellular respiration, and I need to remove carbon dioxide and nitrogen as a waste product. So I want those to easily move across my membrane. Those would be like my air, my oxygen, my air in my chicken coop, okay? Small uncharged polar molecules such as water or ammonia or glycerol are going to mostly move okay across that layer, but there's going to be some that can't, okay? So if I think about back to my chickens, right? I might see some insects move easily across, or even some lizards and things easily move through my chicken wire fence, but some of them can't. Maybe the large butterflies can't move across that chicken wire fence. That's okay. They'll get repelled, but most things move across it without any sort of issues. Okay, my large uncharged polar molecules will be things like my chickens. I want them to be repelled. I want them to stay in the coop. But we all know that there's always one escape artist of some kind. Maybe there was a little hole and a smaller chicken got out or something. We might see that. But we're going to see that mostly those chickens are repelled to stay inside of the coop. Okay, and then my ions, so that's going to be things like glucose and sucrose. Those are going to need a lot of help to get moved across the membrane. And then ions such as sodium, calcium, and chloride generally do not move across that plasma membrane. We want to keep those inside for our functionality. And again, this is going to be my large adult chickens, or if I'm thinking it from the other way, this is going to be keeping those large predators out of my chicken coop. When we talk about the plasma membrane, it's going to want to be permeable to some things and not to others. But we also do have more than just the phospholipids in there. Our lipid membrane has a lot of different proteins embedded in it that are going to facilitate the movement of some of these molecules, even if we think we might not want them moved. Okay? And what that brings us to is concentration gradients. Okay? Nature hates concentration gradients. A concentration gradient is a difference in concentration of substances within a defined space. So basically what this means is that I can have a high concentration on one side, and nature would really like for either side of whatever that barrier is to be equal. Okay, nature wants them to be equal. As such, we generally see if the membrane is permeable to that substrate, we see a net movement towards equilibrium. We see a movement where those molecules will eventually be the same on either side of the barrier. Okay? This is going to be something that plays a huge role in our understanding of passive and active transport, as well as our understanding of building ATP through photosynthesis and cellular respiration. So let's talk about the different kinds of passive transport. So, in passive transport, remember, we don't need any energy, okay? Diffusion is going to be the movement of molecules, and they're going to move down their concentration gradients from areas of high concentration to low concentration until an equilibrium is reached, okay? This is one of my favorite pictures of all time. This is included in the McGraw-Hill book. And I love that they are putting crystal dye into water to show equilibrium being achieved over time. Fantastic. Why do the fish have to be there? If anything, the fish almost stir up the water by their movements and are going to act more as facilitated or an active diffusion. But let's just ignore that for now. Anytime you've ever maybe dyed Easter eggs, you've probably put water and then added a dye to that water. That dye will fall into the water. It's often heavier than the water, but over time it will eventually move through the water, and you'll get this diffusion of these dye molecules. And so your water will go from being clear to purple if you've added purple dye, or clear to red if you've added red dye, whatever it might be there. This is going to be very easy. And this is what we see in things of like oxygen. Oxygen is generally going to move from areas of high oxygen concentration to low oxygen concentration. And so that's going to be an easy movement. The barriers don't really affect anything. It just happens. Okay? Passive transport includes both diffusion and osmosis. So what I just showed you is an example of diffusion, where we have the movement of molecules from high concentration to low concentration. And in this case, both solutes and solvents move. Solutes are the substances that are dissolved in a solvent. So in this case, we have these little red dots. Those little red dots might be something like salt, sodium chloride. Sodium chloride is going to dissolve into water to reach its equilibrium, and water then would be the solvent. We talked about how water is the universal solvent in life. Osmosis is a little different. Osmosis is a type of diffusion, but it is specifically on water. And it is the movement of water from high to low solvent concentrations, which means that it's a movement from low to high solute concentrations. Basically, water is able to move where maybe our salt is not. And this is what we see in cells, okay? Cells are going to contain a lot of salt, a lot of ions, a lot of glucose, a lot of solutes. Our cells will then absorb water based on that concentration of solutes inside of the cell. If it cannot be controlled through homeostasis, like actively removing the water by pumping out those vacuoles, like we saw in chapter 4, then we're going to potentially have a problem where the cell might burst. So let's talk about the 3 different kinds of osmosis that we see happening in cells. First, we can have cells in an isotonic solution. Isotonic is going to be an equal solution where there is no net gain or loss of water. Water is still moving across the membrane inside and outside of the cell, but because the concentration of water is the same on the inside of the cell as in the outside, or we could say the concentration of solutes is equal, then we're not going to see any real change in the cells. Animal cells prefer to be in an isotonic solution. Okay, isotonic solutions means that our cells stay normal. If you look at a red blood cell, it's going to be happy, it's going to be healthy. If you're someone who ever uses contact solution is always isotonic to your eyes, because if you don't have an isotonic solution to your eyes, it's going to affect the water concentration inside and outside of your eyes. And so we want that to be isotonic, and where water can move equally inside and outside of the cells. We want it to match. Plant cells are a little less happy in an isotonic environment. And this is because plant cells have 2 things that animal cells do not. Plant cells have that large central vacuole and a cell wall. The large central vacuole really prefers to be super duper full of water, and when that's really full of water, it pushes against the cell wall. In an isotonic environment, we might see cells if plants start to wilt, and so you're going to see your plants kind of maybe looking a little droopy. They're a little less happy than what they would be in something like a hypotonic environment. If you ever struggle with this, I had a teacher friend teach me, hypo, the cells get big and round, O, and then they'll burst. So let's talk about it. Hypotonic solutions have a greater concentration of water outside of the cell than inside of the cell. Because the cell then has a higher salt concentration or solute concentration, the water is going to move into the cells faster than it can be removed from the cells. As such, this can cause cells to swell. Animal cells that swell may lyse, which if you remember, the word lyse means to break or burst. And this is because animal cell membranes get stretched almost too much like a balloon, and if you overinflate a balloon, it will pop. Your red blood cells in a hypotonic solution will eventually pop. This can result in the blood cells looking like there's fewer cells in the blood, and it can result in potentially anemia and then issues with moving oxygen through the body if you are not in the right balance. Now, this is not going to say, I'm not saying this is going to happen, but we could have a mistake where maybe if we talk about isotonic, saline solution that you might get at the doctor's is 0.9%. If a doctor were to give someone a 0.09% saline solution, that could be hypotonic to those cells, okay? Plant cells prefer to be in a hypotonic environment, again, because they have that cell wall and that large central vacuole. It would be as if you were trying to blow up a balloon, and that balloon were inside a shoebox. You could blow up the balloon to a certain size, but if that shoebox was taped down and solid, you could not get the balloon any larger than that wall allows it to be. Because cellulose is so strong and structural, the plant cell cannot get any larger than the cell wall will allow it to be. As such, this is what we consider turgid in plant cells. This is when your plants are strong, you've watered them, they've stopped being droopy, they stand upright, they're looking real good and happy. That's because those plant cells are now in a hypotonic environment. And I will always say it like that. I'm not going to stop because for me, the O means big and round. And so saying it in a particular way helps me remember it, helps you remember it. Okay? There's one more type of osmotic environment we may find cells in, and that would be your hypertonic environment. So where a hypotonic environment is going to cause the cells to swell either to a certain point in plant cells because that cell wall, or expand beyond the cell membrane in an animal cell, hypertonic solutions cause everything to shrink and shrivel. This is going to be where there is more water inside of the cell than outside of the cell. And so your solute concentrations are larger outside of the cell. And so the water inside the cell will move outside of the cell. As such, the cell becomes dehydrated. It will shrink. Animal cells become something called crenellated, where we have these little spiky dews coming off the membrane here. Those are going to cause clots to potentially occur because these membranes can no longer move across each other smoothly. And as red blood cells are moving around in the veins, if you are severely dehydrated, you are at risk of clots. Plants undergo something called plasmolysis, where the cytoplasm itself starts to shrink away from the cell wall because the central vacuole has lost all of its pressure. When the central vacuole is empty, that cell is going to plasmolysize, and we are going to see a shrinking of that cell membrane in the plant cell. This could result in extreme wilting. It could result in plant death, depending on how dramatic your plants are. But some plants are very dramatic, and if they don't have enough water, they will immediately start to wilt. Plant cells tend to bounce back from this fairly easily. Where you give them water, they'll reverse this plasmolysis, oftentimes within 10 to 30 minutes. Animal cells can be a little more difficult to balance because once it gets to a certain size, those cells begin to die, and we might see them undergoing apoptosis and dying themselves, apoptosis being programmed cell death. In any case, nobody likes to be in a hypertonic environment to their cells, and hypertonic environments will result in dehydration and shriveling that occurs. Okay? Two more kinds of transport I want to talk about: active transport and bulk transport. So active transport is something that we're going to see with both photosynthesis and cellular respiration. The cell needs to use energy to move molecules across a concentration gradient. There are times when cells will want to move something and have it be at a higher concentration inside of the membrane rather than outside of the membrane. For instance, cells that are muscle cells will often release some of the calcium and sodium in a way to move those muscles, but we're going to want to reabsorb those in, and eventually we will be putting those against the concentration gradient. Now remember, Mother Nature doesn't like concentration gradients, and so things like ions don't easily move across the membrane. They are blocked from moving across that plasma membrane. But we can use a transport protein and some ATP to trap those molecules and move them into the cell when they might not move effectively on their own. We see this in both the electron transport chain of photosynthesis and cellular respiration, which we'll talk about more in chapter 6 and 7. So keep this in mind. We are going to be creating a concentration gradient and going across that concentration gradient to make it outside of the equilibrium. And then the cells can actually use that to generate more energy. It's crazy. It's so cool. Okay? And the last kind of transport that we need to talk about is bulk transport. A lot of times those macromolecules, as I mentioned earlier, things like your steroids, your hormones, are too large to be moved by transport proteins, but your cells still need them. So they're going to form vesicles. There are two ways that we see this happening. Exocytosis is movement outside of the cell, so moving from the Golgi apparatus to the cell membrane to then be released outside of the cell, potentially into the bloodstream so that it can move around. Or endocytosis, endo meaning in and exo meaning out, cytosis meaning of the cell. So endocytosis is going to be where we find that hormone in its new environment and then pull it into the cell by making a vesicle. This can happen in the plasma membrane. This is also something we see happening in some of our organelles. So endo and exocytosis are bulk transport, moving large molecules that would not move across the membrane on their own. That's it for chapter 5. Make sure you can do this last, check your understanding. Read the sections of the book that correspond and let me know if you have any questions. Thank you. Have a great day.